Greetings to Dr. Rafidah Binti Othman, the lecturer for the Aquaculture Animal Immunology course. Today, we will be presenting a video lab practical 2 covering blood collection, blood smear preparation, staining, and the observation of immune cells.
The group members consist of Rain Cosmos Robert, CATAGE Ebal, ANISAH Binti and Bok, and MARCIA Michael.
The introductions is blood is an important tissue for studying animal health and immune response. In fish and other vertebrates, blood contains red blood cells, white blood cells, and thrombocytes. In fish, red blood cells are nucleated. Thrombocytes are also nucleated and may look similar to small lymphocytes. Therefore, careful observation is needed when identifying different blood cell types. The objective of this practical is perform blood collection from fish using appropriate techniques while ensuring proper fish handling and sample quality. Prepare a high-quality blood smear by applying the correct smear technique to produce a thin. Carry out blood smear staining using the appropriate staining method. Observe and identify immune cells under a light microscope, including lymphocytes, monocytes, neutrophils, thrombocytes, and erythrocytes. This practical session provides a brief overview of fish blood cells and immunity. Blood cells consist of red blood cells, white blood cells, and thrombocytes. Red blood cells function to transport oxygen throughout the body, while white blood cells play a role in defending the body against infection by identifying and destroying pathogens. Thrombocytes assist in the blood clotting process and are also involved in immune responses. Fish immunity is a defense system that protects fish against bacteria, virus, fungi, and parasite. This system comprises innate immunity, which acts rapidly via mucus, skin, and phagocytic cells, as well as adaptive immunity, which produces specific antibodies following exposure to pathogens or vaccines. The materials required for this practical include live tilapia, an anesthetic solution, sterile PBS, phosphate buffered saline, sterile syringes, 1 ml or appropriate size, sterile needles, the anticoagulant ethylene diaminitetracetic acid, EDTA, microscope slides and cover slips, tubes, a hemocytometer, a microscope, right stain or Giemsa stain, and disposable gloves with a lab coat.
Step 1. Blood collection. Before starting the procedure, prepare a clean and sterile workspace to minimize the risk of contamination. Once the tilapia has been fully anesthetized, carefully remove the fish from the anesthetic solution and place it on a soft, moist cloth to protect its body and reduce the risk of injury. Next, identify the caudal vein, which is the standard site for blood collection in fish. Using a sterile syringe and needle, gently puncture the caudal vein and slowly withdraw approximately 50 to 100 microliters of blood, ensuring that a sufficient sample is collected for subsequent analysis. After the blood has been collected, carefully remove the needle and immediately transfer the sample into a clean, properly labeled microcentrifuge or Eppendorf tube for further laboratory procedures. Step 2. Blood smear preparation. The next step is blood smear preparation. First, clean the microscope slide with alcohol and allow it to dry. Cleaning the slide with alcohol and letting it dry is essential to ensure the preparation of a high-quality blood smear and accurate microscopic observation. Place a small drop of the collected blood near one end of the slide. Using a second microscope slide held at a 45-degree angle, spread the drop of blood along the length of the first slide to create a thin smear. Next, allow the smear to air dry completely, as this helps preserve the normal morphology of the blood cells and ensures optimal staining quality for microscopic examination. Step 3. Staining and microscopic examination. Firstly, prepare Giemsa stain solution according to the manufacturer's instructions. Place the blood smear which has been dried and fixed with methanol for 2 minutes on a staining rack or in a staining vessel. This preparation preserves cell morphology and provides an ideal surface for the subsequent staining procedure. Add sufficient Giemsa staining solution to cover the smear and leave it for the recommended staining period of 15 minutes. The next step is rinse the slide gently with distilled water to remove excess stain. Then, allow the slide to air dry completely. After the stained slide has dried completely, next, gently lower a clean cover slip onto the slide at an angle to minimize the formation of air bubbles. Ensure the cover slip is positioned evenly over the smear, allowing the mounting medium to spread uniformly. This step protects the stained specimen, preserves the blood cells for long-term storage, and provides a clear preparation for microscopic examination. Examine the stained smear under a microscope at various magnifications to observe and identify the types of tilapia blood cells, specifically red blood cells and white blood cells, and record their characteristics. Microscopic observation of fish blood smears. Microscopic examination of the red tilapia blood smear revealed the presence of red blood cells, erythrocytes, white blood cells, leukocytes, and thrombocytes, with red blood cells being the most abundant cell type observed in the smear. Result and discussion. Based on the provided microscope image, 3 main types of blood cells can be observed in the red tilapia blood smear. Red blood cells, erythrocytes, are the most abundant cells observed. They appear as numerous small, oval or rounded cells distributed throughout the smear and are responsible for transporting oxygen. White blood cells, leukocytes, are fewer in number and appear larger and darker than the red blood cells. They contain prominent nuclei and are involved in the fish's immune defense against pathogens. Thrombocytes, platelets, are also present and are generally smaller than the other blood cells. They play an important role in blood clotting and wound repair. Overall, the image shows a blood smear containing erythrocytes, leukocytes, and thrombocytes, with erythrocytes being the most numerous cells observed. Next, this is immune function of each leukocyte type in fish. Lymphocytes provide specific immune protection by recognizing foreign antigens and producing antibodies. Monocytes engulf and destroy pathogens and cellular debris through phagocytosis. Neutrophils provide a rapid response to infection by attacking and digesting invading pathogens. Eosinophils are mainly involved in defense against parasites and contribute to inflammatory responses. Basophils release inflammatory mediators that help regulate inflammation and recruit other immune cells. Together, these leukocytes protect fish against bacteria, viruses, fungi, and parasites. In conclusion, the blood smear practical successfully demonstrated the main blood cell types found in red tilapia, including red blood cells, white blood cells, and thrombocytes. The staining and microscopic examination allowed us to observe and distinguish these cells based on their appearance. Through this practical work, we learned the importance of proper blood collection, smear preparation, staining techniques, and careful microscopic observation in identifying fish blood cells and understanding their roles in fish health and immunity. The source references from about hematological reference values and morphology of blood cells in farmed sex reversed male Nile tilapia and investigation of genotoxicity, mutagenicity, and cytotoxicity in erythrocytes of Nile tilapia.